Please take your seats. A remark relevant to the end of today is that for the speakers, session leaders, other people who are invited to this dinner, please do not go wandering off at the end of the session. We're going to have 3 more talks, and then I've been asked to ask you to remain in the room. And then Nate Frizzell is going to come and take us in groups down to the shuttle bar. You know, kind of random walking off into the distance will lead to you not being lost and not able to get on the shuttle bus in there. Okay? All right, so please don't do that. All right, there are my instructions. I'm passing them along. Okay, so now we're going to continue with this excellent imaging session with yet another exciting speaker. Theodore Huppert from University of Pittsburgh is going to tell us about current and future applications of diffuse optics to biomedical imaging, and I'm sure this is going to be great. Please take it away.
All right, thank you, Ron. Yeah, so on the note of random walks, I'm going to be talking about diffusion and light diffusion through thick samples of tissue. So I'm going to talk a little bit about what my lab does, and then kind of give an overview of the topics and current state of the art of diffuse optics. So what actually is diffuse optics? So I think you're all probably familiar with kind of the idea of conventional optics, ray optics. You know, you get these beautiful images, high spatial resolution. The downside is that you're very limited in depth. Once you start talking about very thick samples, particularly turbid samples, that is biological tissue, now you have to deal with scattering. And so the effect of that scattering is it starts to limit not only how deep the light goes through the tissue, but also your knowledge of where the light went. And so when we talk about diffuse optics, we're specifically talking about optics through very thick samples of tissue where we have to deal with multiple scattering events that really start to limit the spatial resolution. The upside, why we are interested in doing that, is that we can actually start to image through very thick samples. So we can actually start to do in vivo imaging, live animals. I'll show a lot of studies where we actually can do imaging of, say, the human brain or muscle physiology, where we're able to actually get depths of tens of centimeters through tissue. So one of the things that we have to deal with, or the main thing that we have to deal with when we talk about diffuse optics, is this concept of scattering. So in biological tissue, say at about 800 nanometers, the persistence length is around 1/10 of a millimeter, which means that light travels about 100 microns to have a 1/e probability of scattering. So that is for something like your hand down at the bottom, the light has scattered many, many, many hundreds, thousands of times passing from one side of your hand to the other. Although the physical distance across, say, my finger might be about 1 centimeter, all that back and forth meandering of the light means that light has actually traveled more like 5, 6 centimeters just in crossing that distance across your finger. And so the idea of optics or spectroscopy, the idea that we're going to actually try to measure concentrations of chromophores, when we deal with this diffuse regime, actually has to now deal with this back and forth scattering. And so the equation down there, that's what's called the modified Beer-Lambert law. The Beer-Lambert law is something you might remember from maybe freshman chemistry. It says that if you have a sample of tissue, or a sample, that you can measure the concentration of what's in that sample if you know the distance across and you make a measurement of the absorption. That the absorption is proportional to the concentration times the distance. Now, the only problem with scattering is now that distance, as I said, is much, much longer than the physical distance you can measure. So specifically, diffuse optics works in what's called the near-infrared window. So you see from the bottom image, you see from the bottom image there that if you shine a white light on your hand glows red. And that's because your body absorbs very little in the red light. This red light is able to actually pass through because it sits in this optical window. Other colors, for example, green, your body absorbs too much of the light so it doesn't actually go deep enough through the tissue. So there's a window of about 600 to 900 nanometers in which the absorption is low enough that we can actually pass light through these centimeters of tissue. And so that's what we mean by the near-infrared window.
It's still scattering.
But it's still highly scattering, but not absorbing. So it can actually take that random walk of 5 centimeters, and at the end of that random walk, there's enough left that you can still measure something. So inside of that near-infrared window, there's a number of chromophores that we're interested in, and a number that we don't really care about. So this is just a list of what contributes in the near-infrared. The ones that we care most about, because they're either functionally interesting or high enough concentrations, are things like hemoglobin. Hemoglobin, there's some exogenous agents like ICG that are FDA approved. We also have scattering events like red blood cell velocities that change functionally, and we can actually derive contrast from things like that. So kind of the basic idea of this kind of idea of diffuse optics goes back to the 100 years ago with this idea of pulse oximetry. The first commercial models of pulse oximeters really came about in the 1970s. And I think everyone is kind of familiar with this idea. This is how I always explain my studies to my subjects when they come into my lab, that basically what I do is kind of similar to pulse oximetry. I shoot light through your finger, and by measuring the changes in that absorption, how red versus how blue your blood is, I can determine how much oxygen is there, and from that determine your pulse and so on. So this technology has been around for about 30 years or so, and it's the same basic idea from what I'm going to call a first generation NIRS or continuous wave system. So these are systems that are cheap. They consist of basically a light source, which is often a continuous wave like LED. So very low cost, tens of dollars at most, and a very simple detector. So we'll have tissue sitting between a light and a detector. We're just measuring the amount of light that passes through and how much that tissue absorbs at different wavelengths. So the advantage of this kind of first generation system is they're very, very cheap. You can buy systems that have dozens, hundreds even of source and light sources and detectors that actually allow you to make arrangements and make many of these measurements. So it's easily scalable. The downside is because the technology is very simple. You're just looking at how much light passes through the tissue. It becomes very hard to calibrate, and because of scattering, even if you calibrated your light sources, you still don't know the distance. So you don't necessarily know the scattering. You don't necessarily know how far that light has traveled to go across your finger. And that really limits your quantitative accuracy. So that limits your ability to actually recover absolute concentrations. So that hasn't stopped us. In the late 1970s, Franz Jobsis published a paper in Science who demonstrated that he could take that same idea of a pulse oximeter, something you'd wear on your finger, and mount it on your head and actually try to measure brain pulse oximetry. And so this is kind of the current generation. This is what's called a Somanetics Invos system. It's a company out of, I think, Michigan that builds these ICU monitors. They're basically they're this first generation NIRS system. The little sensors up there mount on your head. So here it's shown the pediatric model. And basically what it's doing is it's sending light in. That light is going into the tissue, it's diffusing through the tissue, and then you have a detector that's measuring what light came back out. And by increasing the distance between that source and detector pair, the light is on average traveling deeper and deeper. And so you can find an optimal distance which gives you sensitivity to the brain. So you can have these devices that this right here. You can see on the baby's forehead, it's a little, almost like a Band-Aid. It sits in, and inside of that Band-Aid has a source and a detector mounted in it, and it's measuring the brain pulse oximetry. Okay, so this is a technology. It's been around for about 30 years. There's currently about 4 FDA-approved models that are sold in the U.S. They're very popular in ICUs and NICUs, particularly with cardiac units. I know the hospitals here, almost every one of the ICU units have these things. And so that's kind of this simple first generation pulse oximeter. One of the other things that Jobsis showed in his 1977 paper was that you could do this for brain function as well. And so taking that same concept of this source detector pair that's sampling through tissue, if you put it on someone's head and ask them to do a task, say, tap their fingers in this case. The sensors that are actually over that part of the brain, because that light is penetrating through, in this case, the motor cortex, you'll get a signal change, and you can actually use that to do functional imaging. So in the 1977 paper, Jobsis showed that you could actually put a source on one side of the head, a detector on the other. They had the person hyperventilating, and they showed that they could actually measure the changes in blood oxygenation during this hyperventilation events. Now we know that so the Jobsis experiment will probably never get reproduced because the amount of light that you need to get from one side of the head to the other is far above the ANSI standards. So most of what our experiments we do right now are in back reflectance mode. So as you see a probe there, we'll have sources and detectors mounted in a head cap. Those source and detector pairs are measuring different volumes of tissue, so overlapping volumes. Person does a task, and the volumes that are activated with these vascular changes can be reconstructed into images of the brain activity. So over the, so this idea has grown tremendously over the last about 20 years. Up until about the 2000s, this was mostly in the hands of engineering labs that were building these custom instruments. In the early 2000s, we started to see commercial systems. So now there's about 7 companies that actually build these continuous wave systems. And from that, we've seen a lot of growth in terms of both NIH funding and publications. So type of experiments that this is actually good for, this is just a sample of some of the experiments my lab does. These instruments, the advantage of these instruments are that they're portable. So I'll show some slides in a second, but this is an instrument, it's maybe about 3 feet wide or something, and it sits on a cart. We can wheel this around. It's a lot more, it's a lot less invasive than something like MRI. And so the advantage of this type of technology is you can use it for portable imaging. You can use it for child studies. We do a lot of studies of balance and gait, where you'll wear one of these systems on your head. It's fiber optics to the source detectors, and so you can actually move around. So we can study brain activity in a number of different environments that would be close to impossible with something like MRI. So there in the bottom, I guess it's your left, I took one of these instruments to Ethiopia once, took it up a mountain, did brain measurements on top of a mountain. So something would be impossible with an MRI scanner. And so this is just an example of, this is my favorite slide. This is an example of what brain imaging can look like. Honestly, there's no scientific purpose to this. It was just fun. So this was a study one of my grad students did, demonstrating that we could put one of these caps on your head, have you standing doing a mobility task. So in this case, they're actually playing this Nintendo game where they stand on a force plate and move down a hill. And this is what happens in your brain as you play Nintendo. And so we're able to actually take this this data, record these signals from your brain, reconstruct it into images, do analysis to actually look at real-time brain activity, and I can actually make these images in real time as you're skiing down the hill. As a note, you probably do not want to look at your brain as you're skiing down the hill because it's very, very distracting, but we can actually do it. And so, if we do this skiing down the hill several times, we see a very reproducible change in brain activity, particularly in dorsolateral prefrontal. So this is just showing, this is actually my lab. It's a couple blocks down at the University of Pittsburgh, but this is the system we use. It's a 32 channel, 32 source, 32 detector, so about 1,000 different measurement combinations, different source detector pairs. It sits on a cart. We wheel it around the hospital all the time. So we can do studies either in my lab here, or we can do studies, down in these different environments like the vestibular testing labs. Just to show you kind of the current state of the art, if you get a really high density system, so a lot of these source detector pairs, really pack them in and get all these different overlapping measurements like what Joe Culver did from the Washington University, you can actually start to make images that are actually comparable to things like fMRI. So Joe did this study. He had 672 measurements at each of 2 wavelengths. So this very, very, very high density source detector arrangement. And from that was actually able to do retinotopic mapping inside of the MRI and correlate it very nicely with functional MRI. So the plus side of optical is that it's portable. The downside, if I didn't make it clear, is actually you're very limited in depth. So because of this scattering, we're only able to get about 5 to 8 millimeters into the cortex of the adult brain. So in Joe's study here, he was imaging the visual cortex right on the surface. Had he been going after deeper structures, he couldn't have actually seen them. So there's this trade-off. One of the things that you can do to get around that trade-off is you can start to pair optical up with other modalities. So MRI or CT, image modalities that actually provide good spatial resolution. And so what you can do there is you can take optics, which is able to measure, by measuring spectroscopically, you can measure both oxy and deoxyhemoglobin. You can pair that up with structural MRI, and you can use the structural MRI to guide the forward model, to know where the light went. You can use it to model the diffusion, and because now you have that diffusion model, you can actually start to be quantitatively accurate. You can use that to improve your images. And so by pairing optical up with other modalities, we can actually get images that are comparable to things like fMRI. So this is just showing the same experiment with optical versus MRI during a motor task, showing that we're actually able to get very comparable spatial resolution by using structural additional information. And of course, you can take that and you can actually find ways to complement. So you can use MRI methods that measure things like blood flow, and you can pair them up with the optical that gives you both oxy and deoxy, and you can use that to derive new composite metrics like oxygen metabolism that you couldn't actually get from either modality by itself. So by pairing these sorts of functional contrasts, you can actually get more information. On my walkthrough on diffuse optics, my lab particularly focuses on human brain imaging, but there are versions, you can do the same thing for small animals. And so the advantage there is you can actually do small animal imaging through the intact skull without having to sacrifice the animal, useful for longitudinal studies, let's say. So this was a technique that Beth Hillman, now at Columbia, invented, which was called laminar optical tomography. And the idea of laminar optical tomography was the same idea of what we use in the adult, that is a discrete source and detector pair. We're measuring the light that diffused from the source to the detector. But what Beth did was she built a microscopy version of this, where she basically took a confocal microscope. So a confocal microscope takes light, passes it through a pinhole that rejects all the scattering. So it gets the on-axis light. And what Beth did was she moved that pinhole off-axis. And by moving that pinhole off-axis, what you do is you're only collecting the scattering light. And so you can have multiple pinholes that create virtual source detector pairs. And so by using this laminar optical tomographic system, Beth was able to have, I think it was 7 different distances, and then scan the laser light over. And so she's able to get several thousand of source detector pairs. And from this, actually able to then reconstruct a 3-dimensional image of the rat brain. And so compared with conventional microscopy, where you're limited to about 100 microns, Beth's now able to get 3-4 millimeters into the rat's brain with resolution that's a bit below what you get from confocal, but a lot better than what I get in the adult case. So the advantage of those first-generation systems, as everything I've shown up to now, is that they're cheap. They're simple sources, simple detectors. You can build these very scalable systems. But the downside of which is you don't have path length information. So other groups have worked on methods like time domain. So this is a time of flight measurement. Instead of using a continuous wave laser, you now use a pulsed laser. And so you send a pulsed laser light into the tissue. As the light diffuses through the tissue, that pulse spreads over time, and then you have a gated photodetector that measures that temporal point spread function as the light comes out. And from that time of flight measurement, you're now actually measuring that path length. You know the speed of light and you know the time, so therefore you can actually determine how long, or the length that, can't talk today, the length that light has traveled through the tissue. And so now you can actually start to become quantitative in your imaging. The downside of time domain systems is that they're very, very large, very, very expensive. So up until maybe about 2 years ago, we were talking about you know, TISAF, MiTi lasers, very expensive gated photodetectors. A typical setup like this, which is the MGH system from my PhD, you had one laser and one CCD camera. That system cost about $250,000. Nowadays, you can build it for a lot less because there's been some more recent advancements in fast pulsed lasers. But this was a system, I don't know if you can tell from the image, but it was about this big. We called it portable, but only if you had a moving crew with you. And so it was very, very limited. So you had a lot more information, but it came at the cost of both actual physical cost and the size of this instrumentation.
So, question.
Yeah.
Femtosecond lasers, why so short? Because when you're doing that, the spectral width is going to be on the 100 nanometers or so.
Well, it's 2 centimeters per 100 picoseconds of distance. So typically your point spread function is, you know, as you see there, about 4 nanoseconds or 4 nanoseconds or so. You could get away with, and then that's kind of why the price has come down, is because we've gotten more of the kind of that intermediate, you know, 1/10 of a nanosecond type laser, pulse lasers.
Okay, that's the rep rate you're talking about.
Yes.
You're right, okay, because on the previous slide you said femtosecond laser, the slide before this, right?
Yes. So yes. So with these systems, we were pulsing at
short,
very short. I can't remember if it's pico or femto.
Oh, okay.
10 to the minus 12. Which is that?
That's pico.
Pico.
Okay.
All right. Picosecond.
Yeah, that's fine.
Sorry.
That makes sense.
Yeah.
Femto.
Yeah. Femto would be a little overkill.
Yeah, it would. And not only would you get very spectrally broad, and then you have absorption and other things going on. Yeah.
I'm actually a mathematician, so
okay.
so just to throw a slide in there. So you know, while I do all brain imaging, you can also use this type of technology for other tissue types. So there's a number of groups around the world. This was from my postdoc at Dartmouth, but Keith Paulson and Brian Pogue there, their pictures up in the right there, they work on optical mammography. So same idea, now it's pulsed lasers sending it through. They have a ring of source detector pairs that they time multiplex to get all these different, almost like CT type measurements. And from that, they try to actually, so this is a time domain system, and they can actually recover concentrations of oxy and deoxyhemoglobin, lipid, water, cytochrome C, and try to image this in breast tumors. And so their system, they actually work with Philips, their MRI division, to actually build an MRI compatible version of this. So this thing goes inside of the MRI. The woman has an MRI scan where we get the structural image, and then we're adding to it the spectroscopic. They've come up with ways to mathematically fuse those images and shown that it can actually greatly improve your sensitivity, specificity to tumors. So again, kind of the downside of time domain, while it gave a lot of information, very, very expensive. So an intermediate to that is what's called frequency modulated light, or frequency modulated NIRS. And the idea here is, instead of sending a continuous wave or a pulse, what we do is we modulate the light in the megahertz. So we have an amplitude modulated signal that goes in. We then, as we detect the light, we measure both the amplitude and the phase reference to that input. And from that amplitude and phase information, so if we swept all the frequencies, we do a Fourier transform, and that's identical to the time domain. Most of the systems only operate on one or a few frequencies. And so there's a little bit of assumptions and a little bit more calibration than the time domain system. But this can actually give us absorption and scattering information just like the time domain. The advantage of this is it's a lot cheaper to make. You can start to build systems that have many sources. The detectors are still kind of expensive. So this system, this is the one that's in my lab, has 32 laser sources and only 4 detectors because it's really cheap to add more sources onto this. So what this type of intermediate technology allows you to do is build things like this. This is my handheld NIRS sensor. It's literally a men's razor that we took apart and stuck fiber optics into it. And so we're able to move this around. We have telemetry, so we're able to know where we made these measurements from. And the advantage why the men's razor is actually contours around the head very nicely, so it keeps the contact with the skin. And so we're building this right now as a neonate handheld monitor that we'd be able to bring this into the NICU or right after childbirth, move this around the head and actually do a measurement of brain blood flow and oximetry. In theory, we could also get cytochrome C, one of these redox proteins. And if anyone's interested, I threw this in as I was sitting here. If anyone's interested, we're very interested in integrating this with ultrasound to actually get that structural information so we'd actually know how deep the brain is, which would greatly improve our sensitivity here. This is from my PhD lab, but just showing proof of concept. This was the same, not as pretty as my razor, but same idea. They had this sensor, they moved it around the baby's head, and they were actually looking at changes in blood volume and oxygenation over the first several weeks of birth. So it turns out that right after birth, you go through a lot of changes in terms of your blood flow supply to your head as you switch over from fetal hemoglobin to adult hemoglobin, and that's what we were trying to actually measure for the first time. Just because it's kind of cool, one of the projects that I'm working on is to take this system and validate it against MRI. So we've built a neonate head holder. So the neonate goes in there. We have the fiber optics that go over the visual cortex, so the baby sleeps inside of that little sleigh thing. We shove that we stick that very gently into the MRI scanner, and we can do imaging of both the optical and fMRI at the same fMRI and structural MRI at the same time. And so we're doing both, looking at how quantitatively accurate our baseline measurements of the neonate are, and then looking at functional activity. So far, we've only had one baby, but this just shows the time course for the fMRI and the optical overlaid quite nicely. I can guarantee I'm the first person to ever scan a baby inside of the MRI with both NIRS and MRI, so I'm very proud of this slide. And but we're seeing very tight coupling in terms of the temporal correlation between optical and MRI. So it's really saying that that the optical method is valid. That's something that we've known for a decade now in the adult case. So that's kind of the idea of frequency modulated light. So we've temporally modulated the light, but you can also do it spatially. And so David Cuccia, who's at UC Irvine, for his PhD, came up with this concept of using structurally illuminated light. So they do the same thing that I do with the time frequency stuff, but they do it as a spatial frequency. The equations change a bit, but the same concept. By measuring at different spatial frequencies and then modeling that light, you can actually get the same absorption and scattering quantitative information. So this is a technology that David has now spawned a company off of, with the idea that we can actually now measure absorption and scattering on surfaces, so potential applications to like intraoperative surgery, where we can actually image directly onto the brain to look at the boundaries for tumors and so on, with this sorts of spatially resolved, where we can get both amplitude and then depth information. I'll just finish with a new technology that my lab is working on. So all the other technologies were based on absorption. So we're measuring concentrations via absorption, or measuring scattering. One of the things that is interesting that causes scattering, that changes, is blood flow velocity, or red blood cell velocity. So when you have moving scatterers through your sample, you can actually measure this as a contrast. This is the basis of what's called laser speckle imaging. So laser speckle imaging, if you took a coherent light source, shine it on a sample, it comes back, it creates an interference pattern, If that sample is moving, that interference pattern changes over time. And if you measure either the spatial coherence or the temporal autocorrelation, you can actually determine velocity from this. And so this is a kind of a cool technology that was developed about 10 years ago. This is just showing in a rat, you can take the speckle image, do this, calculate the speckle contrast to look at autocorrelation, and you go from the image on the left to the image on the right, which is just night and day, showing the velocity inside the blood vessels. And so this is a technology that's been around for microscopy for many years. It's just starting to go into the biomedical field. This was a very easy example where you can do the speckle contrast directly on an exposed surface like the skin, so you can actually measure velocity or blood flow in the skin. But more recently, we've started to build instruments that can actually do it in the diffusion regime. So just like our adult imaging where we have a source and detector pair, now we can have a source and detector where we're sending coherent light, and by measuring the temporal autocorrelation of the light that comes back out, we can actually determine blood flow. And so this was a proof of concept from Arjun Yod's lab, who's at UPenn, who showed that they could actually measure blood flow in the adult human brain during, this was actually a finger tapping and a hypercapnia experiment. And since then, we've correlated this with MRI to show that we can actually, the method agrees quite nicely with the blood flow response you get with MRI. So that was my overview of diffuse optics. Hopefully you get some sense at least of what the topic actually is. So if there's any questions.
Compelling. Questions, please. Any questions? Mac, do you have a question? There's one.
You mentioned this ultrasound that you want to combine. In what sense? Do you want to modulate the light?
Well, yeah, I mean, there's photoacoustic imaging that I didn't want to talk about because that's not my expertise. What we do for the MRI, as an example, when we do optical MRI, is I use the MRI, I segment it, I then know this is how thick the skin is, this is how thick the CSF is, this is where the brain is. And so I have a layered model which I then feed in to calculate the forward model, where did the light go. And what we're interested in doing is actually using ultrasound in the same way to just get a layered model. So if I knew how deep the brain was, I can then correct the partial volume, I can correct the forward model, and actually then model the light better and actually get more quantitative information. Photoacoustics is a different field, and that's not necessarily what I was thinking because that's a little bit outside of my expertise. But certainly something I'd be interested in.
What I had in mind is that you also could change temporarily the medium itself.
Yes.
Change the density.
Yes.
And
Diffract off.
So that's
combined with.
Yeah, so there's photoacoustics and optophotonics, and that's the optophotonics, right, that you modulate, use the ultrasound wave to modulate the scattering of the medium and then measure that with the light. And that's definitely another route to go with this type of hybrid technology.
So Matt does have a question. I knew you did.
You've shown very nice correlation with the MR results. And looking at your first point on the conclusion slide, it seems to me that you want to head towards a direction where you don't need the MR.
Yeah.
So do you envision, how do you envision getting the registration? I mean, you could do that with CT also.
Yeah, so we do, most of our stuff right now, we do kind of the same way you do EEG or MEG. So we put the NIRS cap, the optical cap on your head. We have a 3D marker that ding, 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 and measure to the fiducials. We then either, as a second session, get a structural MRI, or what we've been doing in the pediatric case is we've been using atlases. So you register that up to the atlas. For most purposes, most people's brains are roughly the same, at least at the 2-centimeter resolution we're talking here. And so we actually do a lot better using an atlas. Of course, if we had the actual structure, we do better. We do the best. But using an atlas is much better than having nothing at all. And so that's how we've been using the MRI, because you're absolutely right, we want to be standalone.
Because it seems like there's a lot of applications, for instance, biofeedback for rehabilitation. I mean, I don't know if you consider those sort of applications, but it seems like you get prompt measurement right away. You change your stimulus or you change some other thing. It's going to be very valuable.
Yeah, I mean, we've done the DARPA thing. We've had funding to do real-time imaging where we've been able to, measure brain activity, analyze that using support vectors, neural networks, whatever, feed that back as an intervention. It's completely possible to do that.
Can I ask what DARPA wanted for?
Learning interventions. So, yeah, classroom-based settings. Sure.
Yeah, so I was wondering.
Any more comments? Yes, Mike.
So if you want to work in source space, can you use the deviation you see from the joint fMRI years?
So.
Great. Any more comments or questions? If not, let's thank